Welcome to video 3, where we're going to talk further about how molecules move across a membrane. Now, the rate at which a molecule moves is specified as what we call a flux, which is nothing more than quantity over time. So if I say it's millimoles of a substance per second, that would be a flux, okay? Fluxes can be calculated by using Fick's law. So the flux is equal to P, the permeability of the molecule, that is a measure of how easily that molecule gets across the membrane, times the area of the membrane, A, times the concentration difference. So the bigger the difference in concentration, the higher it is, let's say, on the outside of the cell relative to the inside of the cell, the bigger the flux, okay? Big differences across the membrane drive bigger fluxes. So I have two membranes here. They're the same area. Okay, so area is the same in both membranes. I have the flux on the y-axis, and I have the concentration difference on the x-axis. And you'll notice that as the difference in concentration across the membrane goes up, the flux goes up. That's this. So the higher the difference in concentration, the higher the flux in both cases. Now you'll notice in one case, we have a lower flux at a given concentration difference than in the other case. So for molecule 2, I have a lower flux at the same concentration difference than molecule 1. So which molecule is most permeant to the membrane? That is, which has the highest permeability P, case 1 or case 2? Well, the answer, obviously, is case 1, because at a given concentration difference, I have a higher flux. So therefore, molecule 1, if A is the same in both cases, and delta C is the case in both same in both cases, if I have a higher flux, I must have a higher permeability to 1 versus 2. Okay, so that's just sort of a typical graphical way of representing permeability. Okay, so in the case of some substances, you don't need any kind of a transporter. You don't need any special way to allow a substance to get across the membrane. These are usually non-polar lipid-soluble solutes. They're soluble in the lipid, and so they get across fairly easily. They're soluble in the lipid of the membrane. The larger the molecule, of course, the slower that flux will be. Some typical substances that you'll use is doctors that get across the membrane with no help. They're lipid soluble. Codeine, xylocaine, novocaine. Basically, a lot of the anesthetics are like this. Alcohol's like this. So the next time you go to the bar, think about the fact that that alcohol is going right into your cells with no help needed whatsoever. Many gases, if not all most gases actually, that we are going to worry about get across: oxygen, carbon dioxide. We'll talk a lot about in the vital gases block. Inhalation, anesthetics, very important to you as a physician, particularly if you're going to be an anesthesiologist or a surgeon. And so these non-polar substances are different from the polar substances, and in particular they're different from ions, which are not that lipid soluble, and therefore they need the help of membrane proteins to get across the membrane. And the most obvious example of this is the case of ion channels, where you have a protein embedded in the membrane, and it has a channel in the middle of it so that ions can get through the channel from one side of the cell to the other side, from one side of the membrane to the other side. Okay, so ion channels, this is basically, you should recognize, a special case of diffusion. You're moving from high concentration to low concentration through the channel. Okay, so this is facilitated sort of diffusion. Of course, how permeable the membrane is going to be to a particular ion is going to depend on a lot of things. The number of channels present is going to be one of those things. More channels means more ways to get across, and so more permeability. The number of ions per second that any channel will allow to get across the membrane, we call this the conductance. The higher the conductance, the higher the permeability. And the more open channels that you find at any one point, the higher the permeability, because you've got more ways to get across the membrane if you have more open channels. So this is a typical example of protein-mediated transport. A generalized channel is represented right here. And this is a form of facilitated diffusion. That is, it doesn't involve the use of any metabolic energy. So basically, facilitated diffusion is just where a substance is moving from high concentration to low concentration through a channel or a transporter. So in the case of our channel, we have here, as I said, this is just a special case of diffusion where the molecule is going, let's say, from outside to inside through a channel here. Now, most channels have gates. And when the gates are open, we say that channel is open because your ion can get through the channel from one side to the other. If the gates are closed, we say that that channel is in the closed state. And that's because when a gate is closed, that means this channel has a conformation such that the channel is not open. We say that it has a closed gate and therefore the ion can't get through. So the more open channels you have, the higher the permeability is. In the case of carriers, that's represented over here. The easiest way to think of a carrier is a protein that's open on one side or the other. And it just changes conformation like this. Open on the outside, open on the inside, open on the outside, open on the inside, open on the outside, open on the inside. And if while it's open on the outside, a substance binds, then when it changes conformation so that it's open on the inside, the substance will unbind, and it's been moved from one side of the membrane to the other. So now it's open to the outside, now it binds, and now it's open on the inside, and it releases your substance S, and you've transported this substance from one side to the other. You should note, particularly, that carriers and transporters in general are much slower than channels when it comes to transporting molecules. Channels typically transport ions in the case in the millions or tens of millions of molecules per second, whereas carriers are typically in the thousands of molecules per second, so a much lower rate of transport. Okay, and that's the first difference that you should note. You should also notice that whether we're talking about carriers, channels, or carriers, facilitated diffusion accomplishes nothing more than regular diffusion would eventually accomplish. In other words, I can have an ion, it's not very lipid soluble, it's not going to get across that membrane very easily, but if I give it a million years, enough ions will cross to the point where the concentration on the outside will be equal to the concentration on the inside. It's just that if I have a channel and it's open, that will happen a lot more quickly because the substance can get through much more easily than if there's no channel there at all. But eventually the substance will get through. It may take a million years, but it'll eventually happen. So you should recognize that this facilitates the process, but it's not driving the process. Okay? Eventually it'll happen either way. Okay. So some characteristics of carrier-mediated transport that you should recognize. The first is the characteristic of specificity. Carriers tend to be specific for one kind of substance or for one group of very closely related substances, closely related structurally. Okay, and that's what we mean by specificity. So a glucose carrier will not typically transport something that's completely different, some carbohydrate that's got a completely different structure, okay? That's what we mean by specificity. Saturation means that there's some maximum level, there's some maximum rate at which a carrier will allow a substance to get through. So typically, particularly if I have a channel, again, if delta C goes up, my flux goes up. And if it's a channel, it's a nice linear relationship, okay? So that the more the concentration difference increases, the more the flux goes up. And it's nice and linear. Well, for transporters, that's not true. Eventually, you get to the point where a transporter is letting molecules through at some maximum rate. In other words, the transporter is moving as fast as it possibly can, and it's binding substances and moving them across the membrane as fast as it possibly can. And just simply adding more and more substance isn't going to make that rate any faster, because the protein just can't operate any faster. When that happens, we say we've gotten to the Vmax, or the transport maximum, which is this dashed line here. And the transport maximum means that I can increase the concentration difference all I want. I can't get beyond that maximum. And that has pathological and physiological relevance. For instance, in the kidney, you get to the point where you can't transport glucose any faster. And that results in glucose in the urine, which you ordinarily wouldn't see. And that's because you've hit the transport maximum for glucose. We'll worry about that another day. But nevertheless, you should recognize that carriers have a transport maximum, and it can become relevant at very high concentrations. Okay. And finally, we have the concept of competition. So this would happen particularly if you have a carrier that transports two different kinds of molecules that are closely related, let's say glucose and fructose. So you have a carrier in the GI tract which carries both of these. And so if you're looking at how much glucose gets across, typically the carrier will transport glucose. But if there's a bunch of fructose there with it, the rate of glucose transport is going to drop, because that carrier is also going to carry fructose. And the more carriers are binding to and transporting fructose, the less carriers are binding to and transporting glucose. So the more fructose I have, the less glucose gets across, because the carrier can only bind one or the other. And so if I have a bunch of fructose there, that means carriers that would ordinarily be carrying glucose can't do that. So this is the concept of competition. We say the fructose and the glucose compete for the same carrier, and that affects their transport rates. Okay. You should know, although I'm calling these characteristics of carrier-mediated transport, channels do display these three characteristics, but they're almost never physiologically relevant or pathophysiologically relevant. For instance, a channel will saturate, and you will get to the point where a channel just won't let any more ions through, no matter how much you increase the concentration gradient. But it happens at such a high concentration of ions, like molar levels, that you never see it in the body, so it's not relevant. So physiologically, yes, these three things exist, but usually they're not very relevant to the function of a channel, whereas these become very relevant to the function of a carrier over the physiological range of concentrations of many substances in the body. Okay, so just to be complete about it, we think of these as characteristics of carrier-mediated transport. Channels also have them, but they're really not very relevant. Okay, so let's talk a little bit about the type of the other type of transport. In this case, we're talking about active transport. So this would not be facilitated diffusion. This is different. Active transport takes place against an electrochemical potential difference. And what that means is that transporters that exhibit active transport can move molecules from low concentration to high concentration. Facilitated diffusion only happens from high concentration to low concentration. This happens when this can pump molecules, use energy in the form of ATP to move molecules from low concentration to high concentration. Okay? Primary active transport is when ATP is directly utilized to move a molecule up its gradient. So we call these ATPases or pumps. Okay? And a very typical example, one that is in most cell membranes, is the sodium-potassium pump, or the sodium-potassium ATPase. This moves sodium from low concentration in the cell to high concentration outside the cell, and it moves potassium from low concentration outside the cell to high concentration inside the cell. Okay? And so basically this is the reason why you have a sodium and potassium gradient. This pump uses ATP to maintain that gradient. and keep the sodium low inside the cell and high outside, for instance. Okay? We have calcium pumps, for instance, in the cell membrane, which move calcium from inside the cell to outside the cell, from low concentration to high concentration. We also have these pumps, or ATPases, in the sarcoplasmic reticulum or the endoplasmic reticulum membrane. They move calcium from low concentration in the cytosol to high concentration in the lumen of the endoplasmic reticulum, or the sarcoplasmic reticulum. Okay? And finally, we also have proton pumps, or hydrogen ion pumps, and these will, for instance, pump protons into the lumen of the stomach to keep the pH very low to aid in digestion. And you also find these in the kidney, and we'll worry about those in the vital fluids block. We'll worry about the stomach in the food to fuel block. All right. So that's primary active transport. We also have what's called secondary active transport. And these don't use ATP directly, but what they do is they use gradients of ions that are set up by primary active transport. So it's secondary because it's not using the ATP directly, it's using an ion gradient that ATP was used to generate. And by far the most common gradient that's used is sodium. So here I have compartment O outside the cell, and I have compartment I inside the cell. So I have high sodium outside, low sodium inside. So sodium wants to go from high concentration to low concentration. And we can use that want to move other substances against their gradient. So I have sodium, it wants to go through. Bam, bam, it wants to, and here's my transporter. It's like the bodyguard, right? So it's trying to move down, but it can't because the bodyguard is in the way. And my transporter is in the way, and it's not going to allow sodium to move down its gradient unless a substance S is also bound with it. Okay? So sodium will bind here, but it can't get across unless S binds. And once they both bind, sodium gets across. It goes from high concentration to low concentration, and it drives the movement of S from low concentration to high concentration. So S moves from low concentration to high concentration because you're using the tendency of sodium to move down its gradient to do that. Okay? It drives the movement of S. So this is obligatory membrane transport, and in this case, we call this a co-transporter because it's moving both molecules in the same direction. In this case, it's moving sodium from high concentration to low concentration, and it's using that gradient to drive S from low concentration to high concentration. Okay? We also have what are called counter-transporters. Once again, I have high sodium on the outside, low sodium on the inside, and I'm going to use that gradient to drive the movement of S up its concentration from inside to outside. In this case, sodium binds here, S binds here. We turn our transporter, sodium is transported from high concentration to low concentration, and we use that tendency, that energy in that gradient to drive the movement of S from low concentration to high concentration, from inside to outside. Counter-transport because they move in opposite directions. Okay? We also have, just to complete our discussion of transport, membrane-mediated transport, we have vesicular-mediated transport. We have exocytosis, where you take something that's maybe stored in a vesicle, a membrane-bound vesicle in the cell. That membrane-bound vesicle fuses with the cell membrane, and as it fuses, that vesicle opens to the outside of the cell. We would say that that is exocytosis, the movement of a substance from inside the cell in the vesicle to outside the cell. And this happens a lot with synaptic transmitters and with hormones, some digestive enzymes. The specifics of that we will worry about in future blogs. We also have the concept of endocytosis, where the plasma membrane surrounds a molecule and forms a vesicle and brings it in. So now the molecule is in the vesicle inside the cell. So it goes from outside the cell You surround it into the vesicle pinches off, and now it's inside the cell. That's endocytosis. And that is very commonly done through receptor mediation. That is, the substance actually binds to a receptor on the membrane and is stuck to the membrane, and now the membrane invaginates and you form a vesicle and you bring that substance into the body. Iron is an excellent example of that, where it binds a receptor on the outside of the cell, and then the cell membrane envelops it and forms a vesicle that pinches off and brings it into the cell. Cholesterol is brought in by endocytosis as well. Okay. So let's end that discussion of membrane-mediated transport there, and I'm going to wrap up with a discussion of osmosis in the next video, and then after that we're going to do a few problems of a type that I expect you to be able to do for the exam. Thank you.